Thanks very much, Bruce and Tim, for having me here today. I'm going to try to tell you a little bit about MDS, myelodysplastic syndromes. I think about half the talk or a third of the talk, I'll talk about genetics and some of the questions that just came up about how you use genetics in clinical practice. And then the second part of the talk about a class of drugs that includes lenalidomide and thalidomide and how they work and how we might be able to make new drugs. These aren't necessarily exactly relevant to what I'm going to talk about, but Celgene does own lenalidomide, and I'll tell you about the mechanism of lenalidomide. So just wanted to start, since the other talks were more about AML, what defines MDS. The mutations that occur in MDS must cause a clonal advantage for some cell that has the capability of self-renewal that gains a clonal dominance in the bone marrow. So the stem cells with MDS have to dominate the bone marrow, outgrow the other stem cells. Currently, the disease is diagnosed by pathological examination of the bone marrow, and so those cells have to differentiate abnormally and have evidence of dysplasia, morphologic evidence of dysplasia. And that's actually the biggest problem with this disease, is that it's very difficult to do that, to find the subtle morphologic abnormalities. You can see some of those abnormalities in normal people, and different pathologists may see different degrees of dysplasia. So that's something that maybe we can improve on with the use of genetics. And then those mutations that cause dysplasia cause abnormal differentiation, and those cells don't successfully complete the differentiation process, and they ultimately end up with a deficiency of some of the terminally differentiated cells, including anemia, thrombocytopenia, and neutropenia. So those are the defining features of MDS, and really just the difference between MDS and AML is defined by a random number of blasts in the bone marrow, but genetically they probably form more of a continuum. So I'm going to just tell you 3 brief vignettes about the use of genetics for the study of MDS or myeloid malignancies, and then move on to the next topic. So the first is prognosis. So one of the big questions in MDS in a new patient who comes in is what their prognosis is, because our therapy is risk adapted all the way from a bone marrow transplantation to more supportive care. We've gone at this by looking at what genes are focused studies of the genes that are recurrently mutated in MDS in large sample sets. This was a few years ago where we looked at about 450 patient samples. We're now involved in studies that are reaching into the many thousands of samples to really tease this out. But as Tim showed you, these plots show genes on the horizontal axis, and individual patients are vertically, so you can see combinations of mutations vertically, what mutations co-occur and what mutations don't co-occur. And when we did this, we didn't know about splicing factor mutations, but some of the other core mutations were already discovered. And our main goal in this very first study was to see, do these mutations help us clinically at all? We know that these mutations, we could find very powerful associations of these mutations with particular cytopenias, with number of blasts, and all the features that we actually already use clinically to determine prognosis. So you could say that, well, the IPSS score, the International Prognostic Scoring System, which we use routinely for determining prognosis in MDS, maybe that captures all the information that genetics does just indirectly. And when we looked at that, we found in our 450 patients or so, age predicts survival, as of course. The risk groups defined by the International Prognostic Scoring System predict survival in our data set very nicely. And in fact, we've done this now with each of the other prognostic scoring systems that have been developed in MDS, the WHO, MD Anderson, a revised IPSS score. They all predict survival very nicely in our data set. But individual mutations here predict overall survival independent of that of those clinical variables. So mutations in p53, EZH2, and other genes predict survival independent of all the existing prognostic scoring systems, indicating that actually getting genetic information on our patients could actually improve how we predict prognosis in MDS. And this occurs in a substantial number of cases. In about a third of cases, we would change the prognosis of a patient. And they do so in different ways. Patients with p53 mutations are already in the high-risk category. They have high blast counts, thrombocytopenia, complex cytogenetics, all the clinical features that predict poor prognosis, but they still do worse than we would predict by high-risk MDS, whereas these EZH2 mutations occur in low-risk MDS patients that we would normally treat just with supportive care or lower intensity therapy, but actually have higher risk disease and a shorter survival than would be predicted by their clinical phenotype. So one of the big questions for cancer genetics in general is how we take this really complicated genetic information and introduce it into clinical practice, where clinicians may be seeing many different types of cancer. As Tim mentioned, there are scores of genes that are mutated in each subtype of cancer. How can any clinician keep all these things straight and predict prognosis? One potential way is just to create very complicated models and web-based systems or something to help integrate this information, or use pathologists, molecular pathologists to help use this information. One way that we've thought was just to group good and bad mutations together, just as we've actually ultimately done with cytogenetics in myeloid malignancies. There's either good cytogenetics, bad cytogenetics, intermediate. And so what we did here was we took the IPSS score in our data set, and that separated the survival curves for our MDS patients. And then we took, so if you just look at low-risk MDS patients and you say, do you have one of the mutations in one of the 5 genes that predicted a poor outcome, does that change prognosis in these low-risk patients? And it does. It splits that curve quite significantly. And actually what happens is that the low-risk MDS patients with one of those mutations behave like intermediate 1 risk, one group up. If you look at intermediate 1 risk MDS, with one of the bad mutations, they behave like intermediate 2 risk MDS, intermediate 2 risk MDS with a high risk mutation, they behave like high risk MDS. So one relatively simplistic way of introducing mutations into clinical practice is just to group them together, of the mutations that predict a poor prognosis, and integrate that in with clinical variables. We're now working with the group that created the IPSS score, the International Working Group for Prognostic Models, prognostic models in MDS, to create a molecular classification system to integrate this clinical and genetic data into a unified scoring system that hopefully in the next couple of years we'll come up with. So what we like even more than predicting overall survival is predicting therapeutic response, so we could decide how to treat any individual patient. We've done this for azacitidine, one of the indocyanine hypomethylating agents that are useful in MDS. We know with lenalidomide that deletions of chromosome 5q are powerful predictors of response. But one story I'll tell you about here is bone marrow transplantation, which is the only curative therapy for MDS. And can mutations help decide which are the patients that we should treat with bone marrow transplantation for MDS? So we looked at about 90 consecutive patients who were transplanted for MDS at the Dana-Farber and Brigham and Women's Hospital, and looked at about 90 mutations in those cases, and to see what's mutated and associate that with outcome. We found that patients with p53 mutations here did extraordinarily poorly. So their survival is under 6 months with a bone marrow transplant. So you could argue whether the toxicity, cost, morbidity of that therapy is really worth it for somebody who has a p53 mutation, or we need to do something different about how we do transplants in that group. Patients with DNMT3A and TET2 mutations were somewhat intermediate. But the good news was that patients who didn't have any of those mutations actually did quite well, with a lot of long-term survivors. And since many patients, or the vast majority of patients with MDS, are borderline for transplant based on age or donor, this might push some patients without these mutations more towards getting a transplant, and then some patients who might be eligible but has one of those high-risk mutations might think twice about it. We're now, to validate this, this was only in 90 patients, have gone to the National Registry, so we're now sequencing 1,700 patients who were transplanted for MDS around the country and doing this analysis. So hopefully we can make a definitive statement about who should be transplanted based on genetics. So then the last one I wanted to tell you very briefly about is what Tim started telling you about, is this possible premalignant state in the blood. So we came at this. This is a story that has been studied for long ago. Tim gave a very nice background. Actually, Gary Gilliland's first noted that a significant percentage of elderly women who you can analyze by X inactivation studies had evidence of clonal hematopoiesis. And Ross looked at those same group, that these women who had clonal skewing in their hematopoietic system. Actually, some had TET2 mutations. we went at a sort of a data mining study where we looked at exome sequencing study from over 17,000 individuals who had had exome sequencing done as part of various GWAS studies for diabetes or cardiovascular disease or other GWAS studies, and were either cases or controls for those studies. But all these GWAS studies are done with peripheral blood. It's the most accessible DNA you can get from a human. So we looked and all those studies exclude anything that looks somatic because they're really looking for germline predisposition. We did the opposite and excluded things that looked like they were germline and looked for somatic mutations in the genes that are recurrently mutated in hematologic malignancies. And we found that these mutations are quite rare in younger individuals in their 20s and 30s, but starts creeping up in the 40s, and by the time we get to 80, 90, we had a number of patients who were over 100 years old. The numbers can even exceed 20% of all individuals have evidence of a clone in their bone marrow or in their peripheral blood with a mutation in one of the genes that's recurrently mutated in hematologic malignancy. And these are substantial clones. We were only detecting clones that were present in about 5% or more of the cells. So what's this? So first we looked at what genes are mutated. It was not the same distribution, actually, that we see in MDS, nor in AML, nor in any lymphoid diseases. DNMT3A really dominated this, followed by TET2, and then ASXL1. It's actually quite a substantial number of P53 mutations, JAK2, SF3B1, and others. One thing that's interesting, I think, is that almost all of these cases had only one mutation. We know that cancer in general is caused by the sequential acquisition of driver mutations that lead to the full-blown malignancy. This, I think, is really the first step along that road where only one gene is mutated and the others haven't yet fallen along. This is a distribution. We see the same distribution for each of the types of mutations, but the distribution of the allele frequency. So on the order of about 12% of the reads had the mutant allele in these samples. That in almost all these cases, these are likely heterozygous mutation. That means that on the order of 24-25% of all the cells in the peripheral blood bear these mutations, and these are unsorted cells. Many of the cells in our blood are memory T and B cells, which haven't been hematopoietic stem cells for many years. So these actually account for quite a large percentage of the myeloid cells in the peripheral blood. These are completely unsorted, so they'll contain neutrophils, monocytes, and all the lymphocytes. So it's quite a significant number. It's also kind of interesting that that number isn't even greater. It may be because many of the other cells are lymphocytes and don't have the clone, but it may be just that with these single mutations, the clone is not powerful enough to completely take over hematopoiesis, and the normal cells push back to a greater extent and prevent the clone from really completely taking over. And this distribution is actually remarkably consistent. There will be our work will be coming out in the New England Journal in about 2 weeks, back to back with another paper that has this really almost identical findings, as well as the paper that Tim alluded to that just came online at Nature Medicine. So I think there was a question about what the clinical impact of these clones are. So we had a large number of cases. Not all of them did we have detailed clinical phenotype data relevant to us, but on a significant percentage of the cases, we had data about whether these patients ended up getting hematologic malignancies or other cancers. Interestingly, there was no higher rate of cancer in general in these cases. So having a clone wasn't a marker of having an unstable genome and a general cancer predisposition. In fact, we did a GWAS study within our data to see if there was any germline predisposition to getting a clone, and we didn't find anything. But if you look particularly at hematologic malignancy, there's a dramatic increase in the risk of getting a hematologic malignancy, including lymphoid and myeloid malignancies. For all cases, it was about a 10-fold increased risk. If you take just the cases that had at least 10% of the cells having a mutation, or 10% allele burden, then it was on the order of a 40 to 50-fold increased risk of hematologic malignancies. So it's a powerful predisposition for hematologic malignancy. Moreover, leaving aside all of the cases that, leaving out and controlling for having a hematologic malignancy, if you look at overall survival, having one of these clones, having a somatic mutation in the blood increases overall mortality And the reason for that isn't entirely clear. What we did actually have was data on cardiovascular disease for many of these patients. And having a clone was in fact associated with an increased risk of cardiovascular disease. Patients with MDS have increased cardiovascular disease, which may just be anemia. But it is noteworthy that cardiovascular disease is caused to a large extent by blood cells, the monocytes, macrophages, vascular inflammation from those cells, and platelets. And the mutations are generally in epigenetic regulators, and so those cells may not behave quite normally in the setting of cardiovascular disease and could predispose, but we're very actively looking at that right now. So those were 3 quick vignettes that I wanted to tell you about the use of genetics in MDS, that we can use this for the prediction of prognosis, and this is already entered into clinical practice. For the prediction of therapeutic response, that TET2 mutations predict sensitivity to hypomethylating agents. I didn't tell you that story. That p53, DNMT3A, and TET2 mutations predict poor outcome following bone marrow transplant. And then this pre-malignant state, which is very common. It's age-related. We think it's pre-malignant because of the high rate of development of hematologic malignancy, though we haven't in many cases been able to show that the same clone is in the hematologic malignancy that develops just from sample availability. But there's an increased risk of hematologic malignancy and increase in overall mortality. So now I wanted to change gears a fair bit and talk about a class of drugs that are known as immunomodulatory agents that include thalidomide and other drugs that turn out to be extremely useful for the treatment of MDS, and work we've done to figure out how they work. So thalidomide is one of the more extraordinary stories in the history of drug development. As I think you probably all know, it was developed in post-war Germany by a company called Grunenthal, and was developed as a sedative and also an antiemetic, and was brought into clinical practice for the treatment of morning sickness in pregnant women, and was approved in Europe, was approved in Canada for the treatment of morning sickness. In the United States, there was a woman at the FDA named Frances Kelsey who said, under huge pressure from pharma to approve this drug because it had been approved in Europe and in Canada, she said, I don't think that there's enough data in pregnancy to warrant FDA approval for this, and really through her actions prevented children from being born with birth defects from thalidomide in the United States. But became clear quite soon after it was introduced into clinical practice that children that were born to mothers who had taken thalidomide during pregnancy were born with limb bud defects called phocomelia, and some did not survive, some did, and so it was quickly taken off the market. But subsequent to that, the research with this continued. It was found to be a very powerful TNF-alpha inhibitor, found to be effective in erythema nodosum, and then a lab in Boston, in Judah Folkman's lab, put it on some cells and found thalidomide to have anti-angiogenic properties, and based on that finding, started using the drug in clinical trials in cancer, really because of an anti-angiogenic activity. That was a very weak activity, and it's not clear that that was related to any therapeutic responses in humans, but got the ball rolling to do a whole slew of clinical trials in cancer. And the one disease that it actually had marked activity was multiple myeloma, though the reason for that was unclear. But that led all the way to FDA approval of thalidomide for multiple myeloma. Celgene developed 2 derivatives that are very, very closely related to thalidomide, really just a couple side groups that are different from thalidomide, but orders of magnitude more potent on assays of TNF-alpha inhibition, for example, and IL-2 release, again, not knowing whether those were related to their therapeutic responses, but brought those into clinical trials, and this has led lenalidomide to be approved for multiple myeloma. And the reason we started to study it was that patients with myelodysplastic syndrome with deletions of chromosome 5q had an extraordinary sensitivity to the drug, with about 50% of patients having complete cytogenetic responses, as well as mantle cell lymphoma and other B-cell neoplasms that have had some degree of responses to lenalidomide, and pomalidomide, which is even more potent in some of these assays, has now been FDA approved for multiple myeloma. So what we first did was we made a derivative of lenalidomide that could be immobilized to a bead that we could pull down and do mass spec. And when we did this, we found this protein cereblon and DDB1 as direct binding partners for lenalidomide, and that these proteins form an E3 ubiquitin ligase with COL4A and ROC1. As we were doing this, there was another group in Japan led by Hanada who also did a very similar study to what we did with lenalidomide with thalidomide, and they came up with the exact same multi-protein complex. So two different compounds with similar activities, searching the entire proteome, came up with the same protein, so that seemed reassuring. And it also seemed an interesting type of target, both because an E3 ubiquitin ligase has never been targeted by a drug before, but also because E3 ubiquitin ligase targets, and E3 ubiquitin ligases in general make up actually quite a large percentage of our genome. About 7% of all genes are components of these complexes, and they regulate the destruction of very specific proteins. And a drug that binds to an E3 ligase could potentially have many different effects depending on what proteins are degraded by that enzyme. And the effects of lenalidomide and thalidomide are quite diverse. There are sedative effects and antiemetic effects. That's why thalidomide was developed. Teratogenetic effects, the phocomelia from thalidomide, anti-angiogenic effects, the effects in B-cell neoplasms, including multiple myeloma. These drugs induce the release of IL-2 while inhibiting the release of TNF-alpha. These effects are why the drugs are called immunomodulatory agents, and are very, very powerful effects in vitro. And then the activity in del(5q) MDS that we were originally interested in. So could lenalidomide lead to all of these different effects by modulating one ubiquitin ligase? So the assumption was we found an enzyme that is bound by a drug, and so the assumption was that we'd found an inhibitor of this enzyme. So we wanted to know what proteins are degraded in response to the drug, and we again went back to proteomics and did SILAC-based quantitative proteomics to look at all of the proteins that change their ubiquitination in response to the drug. And we found a collection of peptides that are reproducibly have altered ubiquitination in response to the drug, and they almost all belong to just two proteins, IKZF1 and IKZF3, which was quite statistically significant. Then we asked what proteins have altered abundance. This is an altered ubiquitination, which targets cells for destruction by the proteasome. What if we look at the levels of all proteins in a cell? And again, we found the same two proteins have powerfully altered abundance after treatment with the drug. Then finally, we looked at what is binding to cereblon, or CRBN, the protein that actually binds the compound lenalidomide. And we found all the components of the ubiquitin ligase. So the pull down worked, but we actually also found IKZF1 and IKZF3 actually binding to CRBN. And CRBN is actually the substrate adapter for the ubiquitin ligase. Then we did the same experiment with lenalidomide, and the binding of IKZF1 and 3 actually went up. So in the presence of the drug, these substrates were binding even more. So this seemed very interesting. We're getting very reproducible results across multiple different types of proteomic studies. The problem was that it was the exact opposite result from what we expected. We thought we had an inhibitor of an enzyme, which is how most drugs work, of course. In this case, we had adding a drug was decreasing the abundance of these proteins rather than increasing. If we were inhibiting a ubiquitin ligase that degrades proteins, you'd expect their abundance to go up. We found nothing reproducibly going up, only these things going down. So we were a little bit confused, but we could validate these results. So if we look at Western blots for these two proteins, IKZF1 and IKZF3, and we add lenalidomide in increasing concentrations in two different cell lines, these proteins go away relative to a control. And that's remarkable because these are transcription factors, and those are types of molecules that we think about as kind of undruggable. We don't have good ways of drugging them, but we just add the compound, and it turns out in just a couple hours, these proteins start to go away. Same is true in myeloma patient samples that are treated with lenalidomide. The abundance of these proteins goes away. And the activity of these various drugs in myeloma and their potency is recapitulated in their loss of expression of these proteins. So pomalidomide is the most potent drug, and low concentrations, just 10 nanomolar, we're getting a decrease in IKZF3 abundance, and by 100 nanomolar, it's all gone. Lenalidomide is slightly less potent, and thalidomide is the weakest drug. And so the degradation of these proteins mimics their clinical efficacy and their potency in other assays. We then wanted to make sure that we're really altering ubiquitination, and this is how we learned how to do this, but this smear is the polyubiquitination of a protein that targets cells for degradation. So you can pull down IKZF1 or 3 and then stain for the ubiquitin group, and you can see that addition of lenalidomide causes an increase in this smear of polyubiquitination. And we could actually recapitulate that. This is in vivo in the ubiquitin ligase world, means in cells, and in vitro in ubiquitin ligase world means in a test tube. So if we recapitulate all the components of this reaction in an in vitro reaction and then add the drug, we can actually increase ubiquitination of these target proteins in response to the drug. So then we took these targets, IKZF1 and 3, and said, can we find just a little domain in that protein that targets these transcription factors for degradation only in the presence of the drug? And so we made a variety of mutants and found that this little piece here was sufficient to cause lenalidomide-dependent degradation of another protein. So you could stick this little sequence onto GFP or luciferase and add the drug, and then GFP or luciferase would go away. And I didn't show you all of the details, but we narrowed it down to a single amino acid that mediated that interaction. So this is where we were at that stage. We found that lenalidomide was binding to this protein cereblon or CRBN, which is part of an E3 ubiquitin ligase. When the drug is bound, we have increased affinity for IKZF1 and 3 that gets ubiquitinated, targeting these proteins for degradation through the proteasome. And we found that IKZF1 and IKZF3 are essential for the survival of multiple myeloma cells. They're lymphoid transcription factors that myeloma cells are addicted to, and their loss leads to killing of the myeloma cells. And they are also key regulators of IL-2 expression, and so this effect also explains the effects of these drugs on interleukin-2 induction. So this is a fast-moving field. This was we published our paper just earlier in 2014, and then a little while afterwards, crystal structure data was reported by two groups. Our studies, I should say, were also found by Bill Kalin's lab as well as a Celgene group with very, very similar results in the degradation of IKZF1 and 3 in response to lenalidomide. But two groups, this one and Celgene, both solved the crystal structure of lenalidomide and thalidomide bound to cereblon and DDB1. And what you can see is that lenalidomide and pomalidomide as well as thalidomide have part of their molecules that are absolutely identical. It's called a glutarimide ring. And that glutarimide ring makes many different contacts with cereblon, with the protein, and sits in this hydrophobic pocket. So this part, which is the same between all of the members of this class of drugs, sits deep within cereblon. This half of the molecule varies slightly. This carbonyl group is just, is the one difference between lenalidomide and pomalidomide. And that part of the molecule is solvent exposed. It sits outside the molecule, exposing itself. And what turns out to be the case is where it's exposed is right where the substrates are binding. So you've got a drug that sits within cereblon and binds very specifically, but it actually alters the surface of this protein-protein interaction. And that makes a lot of sense to me. It seemed a little too extraordinary that a tiny molecule like this could be mediating the specificity of very specific proteins to be degraded and not other proteins. But it's taking advantage of a broad protein-protein interaction that happens between cereblon and IKZF1 or IKZF3, and then it's just modulating this a little bit. And that's the way that very small chemical modifications here can change the substrate specificity. So the reason that we started this line of work in the first place is because my lab is very interested in MDS, less myeloma. And so we wanted to understand why lenalidomide is so effective in this particular subtype of MDS. And IKZF1 and IKZF3 are really predominantly lymphoid transcription factors and are unlikely to be mediating this effect. So del(5q) MDS is actually the first chromosomal abnormality to be associated with a very specific clinical phenotype in cancer in general. And in MDS, it's its own WHO subtype, characterized by a very severe anemia with large red blood cells, a normal elevated platelet count, and a very pathognomonic megakaryocyte morphology, and a relatively good prognosis. But it's associated with a really dramatic response to lenalidomide. As I mentioned, about 50% of patients have complete cytogenetic responses, about 2/3 have hematologic responses, and some of the patients have extremely long-term responses, even a small number who have come off the drug and remained in complete remission. So the genetics of this are that patients have large deletions that are only involving one allele, leaving the other allele entirely intact. There are no genes that have been identified that undergo homozygous inactivation on this, despite genome sequencing and SNP arrays and many, many studies over several decades. So really, it's a disease of haploinsufficiency, loss of multiple genes, of one allele of multiple genes is sufficient to cause the phenotype of this disorder. So we went back to our proteomics. Our previous studies, proteomic studies, were done in a MM1S multiple myeloma cell line. We redid this in a AML cell line, a myeloid cell line, to see if there were different targets. We actually picked up IKZF1 again, but we picked up a new target, casein kinase 1 alpha, that had increased ubiquitination and decreased protein abundance following treatment with lenalidomide. So we thought maybe that would be a new target that would be relevant to del(5q) MDS. What got us a little bit more excited was that casein kinase, out of the whole genome, turns out to be right in the middle of the common deleted region for del(5q) MDS, one of only 40 genes in that region. Moreover, it's one of the small number of genes within that region that has true haploinsufficient expression in del(5q) MDS. Many genes in heterozygous deletions have normal expression because of homeostatic mechanisms. Casein kinase is one that has really 50% of normal expression when you delete one copy. So it's a good candidate for a haploinsufficient gene that might be sensitizing to the drug. So we redid all the studies that we did for IKZF1 and IKZF3, and I won't show you all of them, but when you treat with increasing doses of lenalidomide, you decrease the abundance of casein kinase 1A1 or casein kinase 1 alpha at the protein level, no change at the RNA level. So this is really just an effect on protein degradation, not an effect on RNA, a decreased transcription of the gene. We actually went and made a conditional knockout of casein kinase 1A1 and characterized it. And this is, I'll just show you this one experiment, which is a competitive transplant through 3 rounds of serial transplantation. And we found that heterozygous deletion of casein kinase 1 alpha actually caused a slight advantage over the wild type cells. It fell during the process of transplantation, but then outcompeted again. Whereas homozygous inactivation caused complete loss of competition. And we found that homozygous inactivation of casein kinase 1 alpha caused complete bone marrow failure quite quickly. So this made us very interested that this could be what some at the Broad Institute have recently called a cyclops gene, has been studied for quite some time as a gene that, when heterozygously deleted, might sensitize to treatment, to targeting that particular protein. Or inhibiting that protein. So here, if you've got heterozygous loss, the cells are just fine. But if you inhibit this even further, the cells can't survive. And so if you take a normal cell and inhibit or degrade casein kinase by half, you'd only be going from here to here. If you took a cell that's already haploinsufficient and degrade it further, you're going towards here and might get a therapeutic window of treating del(5q) MDS cells with lenalidomide. It turns out that we actually had independently found casein kinase 1 alpha as a therapeutic target in an RNAi screen we had done. We did an in vivo shRNA screen a couple years ago that highlighted casein kinase 1 alpha as a therapeutic target in AML, in both shRNAs targeting casein kinase as well as a small molecule that inhibits casein kinase, not selective for 1 alpha, but was an inhibitor. Selectively killed the leukemia cells relative to normal cells. And casein kinase has a couple targets, one of which is beta-catenin. We think that the effects on beta-catenin may mediate that clonal dominance. The other is that it loss of casein kinase activates P53. So if you make a P53 null leukemia and then try to treat it with the drug's totally doesn't affect the leukemia at all. When we looked at the effects of the shRNAs and what they're regulating, we saw a very strong induction of P53. And by Western blot, you could see a strong induction of P53 and its downstream target P21 when you knock down casein kinase. So we think that that's part of the mechanism of killing cells when you lose casein kinase 1 alpha. And in fact, we know that in del(5q) MDS, patients who respond to lenalidomide and then go on to relapse and develop AML, those patients generally develop mutations in P53, consistent with this mechanism. Similarly, here we could take a leukemia that is sensitive to knockdown with casein kinase 1 alpha. If we make a leukemia in a P53 null background, these are MLL-AF9 leukemias, the shRNAs don't kill the leukemia at all. So this is really a P53 dependent mechanism. So we'd made a knockout mouse. We figured out that that casein kinases should be targeting a haploinsufficient gene. Seemed like we had all the pieces to do the killer experiment to take haploinsufficient cells and treat them with lenalidomide and show an effect. And we did a big in vivo experiment, saw absolutely nothing, no toxicity, no effect, which was a little disappointing. Then we thought, well, we hadn't actually checked to see that mouse cells are responsive to the drug at all. So we took a step back and we looked at BAF3 cells, which are murine leukemia cells, treated with lenalidomide, and saw no degradation of casein kinase 1 alpha at all in the mouse cells. Then we went to the literature, which we should have done before we did anything, and found that there really aren't any papers about thalidomide or lenalidomide doing anything in rodent models. And indeed, the thalidomide catastrophe happened to some degree because when rats were treated with thalidomide, there were no limb-bud defects. In murine models of myeloma, lenalidomide doesn't work. So it really doesn't seem to work in mouse cells. Now we know the target of lenalidomide. We know it binds to this protein called cereblon. So if you take these regular murine BAF3 cells and treat with lenalidomide, casein kinase doesn't affect the levels. If you overexpress the murine form of cereblon, still nothing happens. But if you take the human form, the human cDNA for cereblon, and express it in mouse cells, now the mouse cells respond. So casein kinase is degraded in response to lenalidomide. And then I won't show you all the details, but we mapped by deletions and then point mutations of all the differences in amino acids between human and mouse, and found a single amino acid change between mouse and human that mediates this effect. So if you take the mouse cDNA, make the one amino acid change to the human form, now the mouse cells will respond to the drug. And similarly, if you take the human form and mutate that one amino acid, the cells no longer respond, which is useful now because now we can make a model of mouse, a murine model that responds to the drug. It's also potentially the cause to some degree of the thalidomide catastrophe, because if that amino acid change had been different, then rats would have potentially responded to thalidomide and caused birth defects, and the drug wouldn't have moved forward in clinical use. I don't think I have the data here, but it turns out that that one amino acid change sits right in the surface of binding between the substrate and the substrate adapter, and provides steric block for those interactions. So now we could do the experiment where we could look at haploinsufficiency for casein kinase 1 alpha, expressing this humanized form of cereblon so that the mouse cells will respond to lenalidomide, and the haploinsufficient cells with heterozygous deletion of casein kinase induce P21 and P53 much more dramatically than the wild-type cells, indicating a potential therapeutic window for del(5q) MDS, which are haploinsufficient for this gene. And when we did a competition experiment between wild-type cells and casein kinase heterozygous cells, this is in vitro, and then treated with a drug, the casein kinase 1 alpha heterozygous cells were depleted over time, whereas the wild-type cells were unchanged. And if we mutate P53 and look at P53 heterozygous casein kinase heterozygous cells, those cells no longer are responsive to lenalidomide, consistent with the clinical observation that P53 mutant del(5q) cells no longer respond to the drug. So this is our mechanism now, that lenalidomide binds to cereblon. in the presence of the drug that increases the affinity for casein kinase 1 alpha as well as IKZF1 and 3, resulting in ubiquitination, degradation of those targets, and their efficacy in del(5q) MDS as well as multiple myeloma. And these are the first molecules that modulate the function of an E3 ubiquitin ligase, or at least the first drugs. And we've got early data now that modifying this molecule further will lead to degradation of different substrates, and that there may be a potential now to make new drugs that modulate ubiquitin ligases in different ways. And this has been heavily collaborative work between my lab, the proteomics group at the Broad, with David Altschuler for our clonal hematopoiesis work, colleagues at the Dana-Farber for patient samples. And I thought today I just wanted to highlight that Rafael Behar, who led a lot of the genetics work that I talked about in my lab, as well as Zuzana Tatovo, who's here, have both received career development awards from a grant that Bruce is the PI on and has supported their careers, and Coleman Lindsley as well as myself and others benefited from interactions with Bruce over the years. So thanks very much.
Are there kinase inhibitors with selectivity for casein kinase that might be used in this model?
Yeah, we'd love that. There's this one molecule called D4476 that inhibits casein kinase 1 all isoforms, not just R isoform, and it's not very potent, so it's not really a drug, but it's a nice tool compound for in vitro experiments. But we're very interested in developing selective casein kinase 1 alpha inhibitors now. Yeah, Bruce.
Yeah. There are a whole number of human malignancies which are related to translocations that activate different transcription factors.
Yeah.
Is it potentially possible to design a ubiquitin promoting, ubiquitin ligase promoting activity for each one of these?
That's the billion-dollar question. We'd love to be able to do that, and I think we don't know how special this case is and how generalizable it will be. So one approach would be, and which is being done now, is to take your favorite transcription factor that's important for cancer and do a screen to find what degrades it. Another approach is to take these molecules that actually work and keep making derivatives that cause new biological effects and figure out what proteins they're actually degrading, and then figure out what disease to treat with them. And so we're kind of interested in both of those approaches. I think the other big biological question is cereblon special in this regard, or are there other ubiquitin ligases that could be targeted? And right now, it looks like this is the only one that we can do. One other interesting aspect of this that relates to that is that this mechanism isn't entirely novel. In plants, there's a plant hormone called auxin that does the exact same thing. It binds to a ubiquitin ligase and induces degradation of transcription factors. And that is a hormone that's important for flowering and plant growth and signaling within plants. And so it's incredibly tantalizing that that mechanism evolved in plants, and then lenalidomide and thalidomide tapped into that in mammals. It's conceivable that there are endogenous molecules in mammals that we haven't discovered yet that regulate ubiquitin ligases that we're looking for. But it also indicates that the regulation of ubiquitin ligases by small molecules is something that actually evolved in nature. Otherwise, it's almost too extraordinary a mechanism to have just happened randomly. All right.
But I suppose the flip side is there's something that we'd like to increase expressors.
Yes.
Perhaps by inhibiting E3 ligase.
Yeah. Yeah, so it's, yeah. So yeah, we would love to do that too. It's interesting, our proteomic studies really haven't revealed any substrates that we've really dramatically increased. The structural paper that's published in Nature implied that MEIS2 was an endogenous substrate, and if in the presence of the drug, the abundance of MEIS2 would go up because of blocking its degradation. We actually saw MEIS2 in our data and it didn't go down, so I'm not sure if that's right or not, but maybe we had the wrong cell type. But it should be possible. The other thing is that we have proteasome inhibitors and NET8 inhibitors, NETylation inhibitors that can do some of that as well, though that's very broad, very nonspecific. Yeah.
You can only kill a cell once?
Yeah.
Dr. Chabner taught me that years ago. So in terms of the practical matter of the pharmacology of how this approach could best be used in the treatment of diseases, do you really have to keep covering the target constantly, like you would with a kinase inhibitor, where you really want to keep things suppressed forever? Because it sounds like from what you're saying is that once this process, the cartoon you showed in the end, gets triggered, and then the cell dies because of, it's something that would lend itself to more flexible, like dosing once a week, that sort of thing.
Yeah, I think that's a really interesting point, and I don't think we have all the answers yet, but I guess one issue is that you have to catch the cells at the stage when they are absolutely dependent on IKZF1 and IKZF3 for survival, or casein kinase. So if those cells go through different differentiation states with different degrees of dependence, then that would modulate it. But it's a great point, and I think there are clinical studies ongoing to alter dosing regimens as well. One of the other complicated things that I think this brings up is that we actually use proteasome inhibitors as well as lenalidomide for the treatment of multiple myeloma. These are now antagonizing effects, not helpful effects. The proteasome inhibitor will block the function of lenalidomide, so it will definitely inform the development of dosing regimens of how you use those two drugs for the treatment of myeloma, that you would want them to both at least half kill the cell if you can't kill the cell twice, but not have them fully therapeutic simultaneously.